Assalamu alaikum and welcome everyone to our molecular biology ASCP preparation series. This program is all about providing real life MCQs for your certification success. The MCQs we'll discuss today and in upcoming session have been carefully collected from several years of past exams to give you the best preparation possible. I'm Dr. Laya Abdelati, certified as international technologist in molecular biology, and I'm really excited to share my knowledge and experiences with all of you through this series. I also encourage everyone taking the test to share your question with us so that everyone can benefit. So let's dive in together, but don't forget to subscribe to our channel to receive all the latest lecture. All right, so let's get started. The molecular science part is divided into four sections: nucleic acid chemistry, basic molecular theory, biochemical reagents, and genetics, which together make up 20 to 25% of total molecular biology ACB exam. In the previous lectures, we completed the multiple choice question related to the nucleic acid chemistry, and we moved to the basic molecular theory section. Replication part. Okay, for those who missed the previous lecture, I have added the links to those lecture in the bio below for your referencing, so don't miss to like, share, and subscribe to our channel to get the latest lecture. Right now, let's focus on the question related to the DNA replication in eukaryotic cells and review ASCP questions from the previous years. Okay, so now let's see the first question we have today. What is the direction of DNA synthesis in both leading and lagging strands? Give you 10 seconds. The DNA polymerase can only synthesize DNA in the 5' to 3' direction by adding nucleotide to the 3' end of the growing strand. So the correct answer is 5' to 3' direction. Okay, let's move on. So the next question is, which DNA structure is replicated last during the S phase in eukaryotes? 10 seconds. The correct answer is telomerase. Actually, telomerase located at the end of chromosomes are replicated last due to the lagging strand problem and the role of telomerase. Alright, so let's move on. Okay, the next question is, what is the function of telomerase during DNA replication? Alright. Okay, the function of telomerase, they prevent the loss of important DNA sequence at chromosome ends. Alright, let's move on to the next question. Okay, what role does telomerase play in eukaryotic replication? Alright, the correct answer is extend the telomerase to prevent DNA shortening. Actually, telomerase adds repetitive sequences to the end of chromosome to contract the end of replication problem. Okay, let's move on. Which part of chromosome ensures complete replication of linear DNA in eukaryotes? Alright, the correct answer is telomere. Actually, telomeres are repetitive nucleotide sequences at the end of chromosome, as we see in the image. They protect the ends of chromosome from deterioration or from fusion with neighboring chromosomes. All right? During DNA replication, the enzymes telomerase help to maintaining the length of telomeres, ensuring that the entire chromosome is replicated without what? Losing important genetic information. All right, let's move on. The question is, what is the primary purpose of Okazaki fragments in DNA replication? The correct answer is to allow replication of lagging strand. As we know, Okazaki fragments are short DNA fragments sensitized on lagging strand, all right? Because the DNA polymerase can only sensitize in 5' to 3' direction. Okay? So the correct answer is to allow replication of lagging strand. All right, let's move on. Here is the question. How does histones influence DNA replication in eukaryotic cells? All right. As we know, histones are proteins that package and order the DNA into structural units called nucleosome, right? So, during DNA replication, histone must be temporarily disassembled to allow what? To allow the replication machinery to access the DNA. Alright? After replication, histones reassemble to reform the chromatin structure. Alright? So the correct answer is C. Histone must disassemble ahead of replication fork and reassemble after replication. Alright? Let's move on. All right. The next question is, which of the following is the characteristic of the replication fork in eukaryotic cells? 10 seconds. Let's move on. All right. The correct answer, it has the leading and lagging strands sensitizing in opposite direction. As we know, the replication fork moves in both direction with the continuous sensitized on leading strand and discontinuous sensitized on lagging strand. All right. So let's move on. The next question is, what is the primary role of DNA polymerase alpha in eukaryotic DNA replication? The correct answer is to initiate DNA synthesis by adding the first few nucleotides after the primer. DNA polymerase alpha initiates the DNA synthesis by extending the RNA primer, as we know, right? Providing a short DNA fragments to begin replication. All right. So let's move to the next question. The next question is, which protein complex is responsible for loading the DNA polymerase onto the DNA template in eukaryotic cells? The correct answer is clamp loader complex. The clamp loader complex, also known as the replication factor C, is responsible for loading the sliding clamp onto the DNA template. The sliding clamp then helps to keep the DNA polymerase attached to the DNA, ensuring efficient and processive DNA replication. Alright, let's move on. The question is, What structure is formed when two replication forks meet during DNA replication in eukaryotic cell? All right, let's move on. The correct answer is the replication bubble. All right, a replication bubble forms when the replication forks meet and the DNA is fully replicated between the two forks. All right, so let's move on. Okay, what happens when the DNA polymerase encounters a replication fork barrier? Actually, when a DNA polymerase encounters a replication fork barrier, it typically stalls rather than continuing its synthesis, right? This stalling occurs because the polymerase cannot proceed past the barrier due to the physical obstruction or structure abnormalities in DNA. All right? The inability to move forward effectively halts the replication process at the point. So the correct answer, the polymerase stalls and replication stops. All right, let's move on. The next question is, which of the following is true about eukaryotic replication origins? The correct answer is multiple origins of replication are activated during S phase. In fact, eukaryotic chromosomes have multiple origins of replication that are activated to ensure that the replication occurs quickly and efficiently across the entire genome. All right, so let's move to the next question. Here is the next question. Which eukaryotic DNA polymerase is primarily involved in DNA repair mechanism and replication restart at neck and lesions? The correct answer is C. DNA polymerase beta is essential for maintaining genomic integrity by repairing single-strand breaks and other forms of damage that can occur due to the environmental factors or normal cellular metabolism. All right, let's move to the next question. Which eukaryotic DNA polymerase is primarily involved in repair mechanism but can substitute for DNA polymerase delta when needed? The correct answer is DNA polymerase beta. DNA polymerase beta is eukaryotic polymerase primarily involved in base excision repair. All right, a mechanism for repairing damaged bases in DNA. While its main role it is in repair, but under certain conditions it can perform function complementary to DNA polymerase delta, particularly in filling gaps during repairing process. This versatility allows to act as a substitute for DNA polymerase delta in a specific context. Alright, let's move to the next question. Okay, the next question is, what is the rate of mammalian DNA replication? The rate of DNA replication in mammalian cell is approximately 50 nucleotides per second. Therefore, the correct answer is C. Let's move on. Which eukaryotic DNA polymerase is primarily responsible for sensitizing the leading strand during DNA replication? Actually, in eukaryotic cells, DNA polymerase epsilons is primarily responsible for synthesizing the leading strand during DNA replication. It works alongside other enzymes such as DNA helicase, primase, and DNA ligase to ensure that the leading strand is synthesizing smoothly and without any error. Okay, so the correct answer is DNA polymerase epsilons. Let's move to the next question. Here is the question. Which eukaryotic DNA polymerase is exclusively responsible for mitochondrial replication? The eukaryotic DNA polymerase exclusively responsible for mitochondrial replication is polymerase gamma. Gamma, alright? This enzyme is found in the mitochondria and play a crucial role in both replication and repair of mitochondrial DNA. Alright? So let's move on. The question is, what is the rate of mutation per round of DNA replication in eukaryotic? The rate of mutation per round of DNA replication in eukaryotic cells is 1 in 1 billion base pairs. This low mutation rate is crucial for maintaining genetic stability cross generation. Alright, let's move on. Which enzyme initiates lagging strand DNA replication by sensitizing RNA primer? The correct answer is DNA primase. DNA primase is the enzyme is responsible for initiating lagging strand DNA replication by sensitizing a short RNA primer. This primer provide the starting point for DNA polymerase to begin sensitizing the new DNA strand. All right. Okay, the next question is, which DNA polymerase is primarily responsible for the elongation of the RNA primer during lagging strand DNA replication in eukaryotic? All right. Which one of the four options below? The correct answer is DNA polymerase delta. Actually, during the replication of lagging strand in eukaryotic cells, primase first sensitize RNA primer, right? Following this step, DNA polymerase delta step in to extend the primer into the longer segment of newly sensitized DNA. Starting from where? Starting at the 3' end of the RNA primer. Then DNA polymerase delta adds nucleotides complementary to the template strand and continues this elongation process until it encounter another RNA primer or a previous sensitized Okazaki fragments. Okay? So the correct answer is DNA polymerase delta. Okay, the next question is, during DNA replication, the 3' hydroxide of the growing DNA chain attacks which phosphate of incoming nucleotide? Alpha, beta, gamma, or delta? The correct answer is alpha. Actually, in DNA replication, the 3' hydroxide group of sugar in the growing DNA chain attacks the alpha phosphate group of the incoming nucleotide. This results in the formation of phosphodiester bond between what? Between the 3' carbon of the sugar of the existing nucleotide and the 5' carbon of the sugar of the incoming nucleotide. leading to the elongation of the DNA strand. All right? Okay, here is the next question. Which of the following is doesn't play a role in DNA replication? The correct answer is guanyl transferase. Guanyl transferase is involved in messenger RNA capping, not in DNA replication. In contrast, DNA polymerase, helicase, and single-strand binding protein are essential for the DNA replication process. All right? Let's move and see the next question. What is the name of the enzyme responsible for the unwinding helical DNA for replication? 10 seconds. The correct answer is helicase. As we know, helicase is enzyme responsible for unwinding the helical structure of DNA during replication. It catalyzes the breaking of the hydrogen bond between the complementary base pairs, allowing the two strands of DNA double helix to separate and form a replication fork. Alright, let's move on. The question is, all of the following enzymes are part of the DNA replication machinery except Okay, the correct answer is luciferase. Luciferase is enzyme that catalyzes the bioluminescence reaction in some organisms and is not involved in DNA replication. The enzymes that are part of DNA replication machinery include ligase, DNA polymerase, and helicase. Alright? So we have to pay attention to the question that contains the word except because it's going to change the meaning of the question. Alright? Here is the last question we have it today. So, what is the difference between topoisomerase and helicase? The correct answer is B. Topoisomerase relieves supercoiling tension while helicase unwind the double helix structure of DNA. Let's explain it in a simple way. As we know, topoisomerase works by what? By cutting and rejoining the DNA strands to relieve the tension caused by supercoiling, which occurs ahead of replication fork, right? In contrast, helicase unwinds the double helix by breaking the hydrogen bond between the base pairs, allowing the replication machinery to access the single strand DNA. Alright? Alright. This table provides overview of key eukaryotic DNA polymerases, highlighting their function and role. Alright? So we need to pay special attention to this section for your exam. Many questions often come from here, so understanding it well can really help you score higher. Alright? That's all for today, guys. Please don't forget to like, comment if you have any questions, share and subscribe to our channel to receive all the latest lecture. Salam.